So the next speaker is Sara Prada, who will speak about topological data analysis of high-dimensional correlation structures with applications in epigenetics.
Okay, can you hear me? Okay, so today I'm going to talk to you about my PhD in mathematics that I finalized by February this year. And I would like to transmit the main ideas, so you will not see a lot of formulas here. But I would like to transmit the main ideas of this work that I think is establishing a link between, or a bridge, between mathematics and biology, specifically about mathematics and genetics. So what we did with this work basically was to propose novel analytical tools, establishing a link between the mathematical thinking or mathematical realization and the biological observation. And that goes very much in line with this article published in Nature by October 2020 called The Forefront of Genomics, where they talk about, or they present, the challenges associated to the analysis of genomic data and how we need to develop novel methodologies. In fact, when we started to treat with genomic datasets, we faced many challenges that were not solved completely with current techniques, as how can we model a dataset with more than 400,000 variables, calculate the correlation matrix and be able to interpret it? How can we create standard analytical tools that can be used not only by mathematicians, but for example, by physicians as a diagnostic tool? And a biological question, how is the epigenetic network working? So we developed several objectives. First of all, as I said, novel analytical tools to study high-dimensional correlation structures with an efficient application and a potential diagnostic power to be used in medical research, understand complex epigenetic mechanisms, and present mathematical strategies that could be used in other research fields. And in order to achieve those objectives and deal with those challenges, we proposed, first of all, a topological approach to treat epigenetic data, to study the high-dimensional correlation matrices through the related correlation networks that we created in different ways, as I will explain. And so we proposed a model and a computational algorithm to fulfill with those objectives. I remember that Rosa said that complex data is not always big, but I think big data is always complex, and genomic data is an example. So the way we did this was using a transversal design that I think is very much in line with my mathematical profile. So we used techniques from algebraic topology, statistics, computer science, and all the background on epigenetics to be able to understand these data sets locally and globally. I would like to give you, first of all, a bit of the biological context that we had to face. So, first question is, what is beyond the DNA sequence? Not everything is coded in the DNA sequence. There are much more complexity levels that we have to take into account in order to be able to propose rational models. So first of all, we have epigenetics, all those genomic modifications that are altering our genome somehow without changing the DNA sequence and are caused by our interaction with the environment. There are different types of epigenomic modifications. We treat one of them called DNA methylation, that is known to have an effect on different levels of methylation that are represented here with these beta levels. They are known to have an effect on how the related genes are expressed or repressed, and therefore, for example, have an effect on the progression of pathogenesis or of diseases as cancer. In fact, there are already approved drugs by the FDA that interact with the methylation levels of the genes in order to avoid or improve a certain disorder. And on the other side, we have the genome architecture. So the way the DNA is grabbed into the nucleus of the cell, it defines itself a level of a structure because it's supposed to join distant sites that are far away in the genome sequence initially, but they become together thanks to these cell forces. So this establishes an structure that is supposed to be non-random, and that has to be understood and modeled in order to understand how this epigenetic network is working. In fact, in a first exploratory analysis, we found significant levels of correlation between sites located in the same and different chromosomes. As you can see here in the circle plot, where its color is a chromosome, and its line is a correlation greater than 0.8. So yeah, conclusion is molecular biology is a mathematical challenge, so mathematicians, we have to investigate that. And the main inspiration, technical inspiration of this work was a new trend on data analysis called topological data analysis that is based on the translation of the data into the language of algebraic topology in order to study the shape of the data and the data invariants that are related to the main data characteristics. There are different techniques of TDA. Persistent homology, for example, is based on homology groups theory. Mapper is based on Moore's theory and the identification of critical points with the construction of the rib graph. And Mapper was very used in the last years for example, to analyze in brain imaging data or neural networks, or in general in medical practice to understand this complex data, and it's based on a clustering procedure. So we start with a data cloud. We map the information of this data cloud into R, but it could be in other dimensions too, with a filter function of interest. And we then apply cluster over the inverse image of those intervals in order to define a network that is supposed to be representing the main characteristics of the data cloud, where its node is a cluster and they are joined if they have common points in this case. So the basis of Mapper and generally of TDA is to let the data speak, doing minimal assumptions about the data space and reducing the dimension of the initial data cloud through the creation of simpler objects that are more manageable. So we start with our work. We separated our work on the study of the local correlation within a chromosome, because it is known it has a different behavior than the global chromosome of sites, epigenetic sites or methylation sites located in different chromosomes. In particular, we studied the aging process as a motivation. We wanted to know how the correlation is evolving through the years and how this can be explaining part of this aging process or the longevity process. So you can see here several correlation matrices with absolute values greater than 0.8 for different sample groups of different ages. These are different individuals, and you can still observe an structure over the red circles and the green circles that are representing the correlation of sites located nearly and sites located far away in the DNA sequence. These matrices are correlation of certain sites of the genome called CPG islands that are very known to have an effect on the gene expression that I mentioned before. So what we decided is to understand, so to describe those structures, creating the associated correlation networks where its node is a site and they are joined as per their Pearson correlation, and understanding or let's say modeling the behavior of those networks in order to be able to describe this correlation design. So with the representation of those networks, we were able to observe an analytical difference on those matrices and a structural difference, for example, with the modularity design that is being represented, and we observe how it's different for some of those age ranges. And the main thing that we did, so we took one of those networks, we applied a filtering, in this case going from higher levels of correlation to lower levels of correlation, and we applied persistent homology in order to observe what were the topological elements that were being formed with each one of the steps of this filtration, in order to first of all differentiate this behavior from a random design. So we were able to demonstrate that this is not a random design, this is not a random behavior. And we were able to demonstrate, thanks to the observation of this and the comparison of these topological elements, how there is a difference between the correlation of sites located more near in the genome sequence and those that are located far away. And this was not done in this way in literature. Literature is very focused only on the sites that are located nearly, but the overall study of how they interact with each other was not totally covered. So the network descriptors were mainly, of course, the correlation, but the distance between the nodes. And that was the main factor we used to propose a model for those networks. In order to model this community structure that were observed, this model was based basically on a couple of matrices, zeta and C. Zeta was defining the blocks that we observed, for example, in the diagonal, and C was defining the probabilities of joining a node in the same block or in a different block. With this model that is basically estimating the adjacency matrix of the networks, we were able to predict, first of all describe and then predict the structures that we observed in the first matrices. And we were able to observe a curious, well, what we did was to reduce the dimension of a matrix that could be 100 per 100 to a matrix C that has, for example, for 4 blocks, it has 10 parameters. So it allows us to observe the evolution of the correlation through the years. And we observed a curious behavior that was that there were higher levels of correlation for very young people, but also for very old people. And we discovered this on different sites of the genome, some of the biggest CPG islands, and we wondered if we were observing the secret of longevity. Of course, we couldn't demonstrate this, but I think it's something for further research. And in order to study the global correlation, so moving forward after the local study, we have defined, or we have designed, a computational algorithm called MultiNet that is kind of an evolution from Mapper because, well, I would say it's a different methodology. It's inspired on Mapper, but has much more functionalities. So Mapper is not very efficient when working with high-dimensional datasets because it's designed to differentiate sample groups and normally forces you to calculate the correlation matrix by brute force, so in the way we normally do it. But as here we have to treat with datasets with more than 400,000 or 800,000 variables, we had to follow a divide and conquer strategy. So we divide our network, our dataset in different windows. So we calculate the correlation matrix and we represent the correlation matrix for each one of them, and afterwards we join everything in order to observe it. I will explain it better afterwards. But we added much more functionalities, so we have more flexibility of joining nodes, we applied more clustering techniques, we follow a local and a global perspective. That is very important when you treat complex data, so to observe all the layers. This algorithm is more robust depending on the parameters you use. Mapper is very unstable. And we are doing future modeling. So we apply clustering over the variables instead of the samples. And instead of presenting here the long code that we have used, I decided to use these pictures where you can observe the main steps of MultiNet. First of all, as I said, we divide the dataset, or in this case the variables, in different windows by a filter function of interest, H, that could be, for example, the median, the variance, the genome position of those sites. We map the information on it contained on each one of the windows in R2, defining the filter functions F and G. That could be, again, median or variance, or the median, the difference in the medians if we are studying different sample groups. And we find overlapping intervals on this mapping. Over each interval, we apply Cummings cluster, and we define a first network that is where each node is a cluster of sites, and they are joined if they have a common point or a high correlation. We do the same for all windows. We select, and afterwards we join all the networks, and we start the process again of joining nodes if they have common points or high correlation for overlapping windows, in order to, because if you do it with windows that are not overlapping, then you destroy the shape of the data. You are not able to observe it anymore. But the process is not stopping here. So we need to understand what this network is telling us, what this network is representing. So we implemented more functionalities to be able to, for example, select the nodes of interest that were describing methylation patterns that were associated with the distinction between the sample groups, and more things that at the end you should extract from those networks that are representing your global correlation structure. This algorithm is public, is in GitHub. Well, the name MultiNet comes from multiple networks, of course, and is able to process the entire array of more than 400,000 variables in less than 50 minutes with a normal computer, which I think is, quite fast. But in any case, normally you don't need to process the entire array, because you do have several ways of, for example, selecting a set of windows where you find interest. And so, for example, if you select the first 5,000 windows, you will obtain your results in 10 minutes. So this is a very simple example on how those networks are representing the data cloud, and with a simple coloring of the nodes, we can observe how this is successfully being represented. This is just a network with 5,000 sites. And if you create your networks for for the sample groups of the aging process study that we did before, with newborns and nonagenarians, we can observe a difference in the layout of the networks, a difference in the distribution that is behind of those networks. With, again, just coloring the nodes, you observe how the quartile distribution is telling us that the oldest group has, in general, lower methylation levels than the younger group. And in order to prove that this algorithm was a successful tool to observe the correlation of the epigenetic mechanisms globally, we applied this algorithm to different datasets of cancer disease. So, for example, using prostate cancer, using colorectal cancer, we were able to demonstrate that we find quick results in line with literature, but let's say that you have them all together. You have a lot of information that you can extract from those networks, so you don't need to be searching for those answers using different techniques. I didn't mention that, but MultiNet has also implemented different statistical models to be able to detect the sites that are significantly associated with the sample distinction. We use regression, random forests in order to detect those sites. And in this heat map here, you can observe how there is, in fact, a difference in the significant sites that we found. In this case, with higher levels of methylation for the cancerous state of patients. And we also observe what is behind of those sites. So where are they located in the genome? What are the genes related? What are the patterns of methylation behind those genes? And of course, when you try to propose predictive models for genomics, you have to reduce your variables, because if you tell a physician that we'll need 5,000 CPG sites or genes to predict a cancerous status, well, they will not use it, basically. So we need to reduce the number of sites and genes that we use as prognostic factors. And we were able, in this case, to propose 10 sites and genes, some of them in line with the proposals in literature that were a very high prediction rate on different datasets of this prostatic cancer status. So, yeah, conclusion is that we were able to test our initial hypothesis of a structural correlation in epigenetics and methylation with novel techniques that we have developed, not new TDA methods, in particular a mathematical model and a computational algorithm designed to describe and predict this correlation design. Again, I would like to emphasize that this was done thanks to the transversal design of using different mathematical techniques from different areas in order to understand this data structure completely and therefore being able to model it successfully. And I think we went very much in the line of multidisciplinary design of generating biological hypotheses from observations that we can solve with mathematics. That is, again, I think very much in line with the current data scenario where you do need multidisciplinary teams in order to understand complex data. And I think one of the good things of this work is that it's establishing more bridges as open research. So, for example, with the real use of the presented techniques, the biological collaboration and interpretation, of course, we need a team of biological experts that are able to understand all results that we provide them. So we provide them with the tools, they have to interpret it. And the extension to other research areas where high-dimensional or big data, complex data can be gathered. And that's all. Thank you.
Any comment or question? Yes, I only one on my part. In epigenetics, typically Pearson correlation is a not good selection sometimes, no? Because, for example, with SNPs, you have 0, 1, 2 categorization. No? Then I am asking about if all these graphical tools, for example, using local correlation, can be also translated to general association measures for genes?
I think so. Yeah. They can be perfectly. So the thing with genomics is that you do have, well, and with data analysis generally, you do have many options, and you have to force yourself to restrict very much what you are doing, because otherwise, for example, the modeling of these SNPs is something that we can say deserves a specific work. So you cannot just take MultiNet and apply to SNPs. MultiNet is more designed to understand those DNA methylation patterns or gene expression patterns. So you will need to adapt your work so you are able to understand and model all the kind of phenomenon or information that we can extract from our genome. But yeah, I think it could be done. Yeah.
Okay. No more comments, so I think thank you to all speakers for this.